So thanks everyone for joining us today. Welcome to MACAN's final webinar for the spring. Today we are talking about seagrasses in the mid-Atlantic, restoration efforts and blue carbon potential. So just some general information about this meeting. We're going to have a questions and comments and answers, of course, at the end. It'll be about 15 to 20 minutes. Everyone is going to be muted during the webinar, and we ask that if you have questions, you can put them in the Q&A box or in the chat, and we will be monitoring as we go. This presentation is being recorded, and it will be available on midacan.org/webinars, and we will have an email go out to all participants as well as the MACAN listserv when the webinar is posted on our website. I also just wanted to bring everyone's attention to the MACAN in-person biennial State of the Science and Technical Workshop, which is July 15th to 17th at the Smithsonian Environmental Research Center in Edgewater, Maryland. The QR code on your screen is for the registration link for that meeting. It is free to attend, but we will have a meeting cap. So we're almost there. So if you are interested in attending, please do register immediately. And finally, this webinar is in collaboration with the Mid-Atlantic Regional Council on the Oceans, or MARCO's Coastal Carbon Collaborative Work Group, which is a work group that is focusing on nature-based solutions to acidification, as well as MCDR. So if you have interest in hearing about that group, please feel free to email info@midatlanticocean.org, and we'll be happy to get you more information about that. So for those of you who may not be familiar with the Mid-Atlantic Coastal Acidification Network, we are a group of scientists, federal, tribal, and state agency representatives, state resource managers, federal resource managers, non-governmental organizations, and other nonprofits, as well as industry partners. We focus our efforts in the geographic region from Long Island Sound to coastal Virginia, and we are one of the 7 coastal acidification networks across the country that is funded by the National Oceanographic and Atmospheric Administration's Ocean Acidification Program. We have goals, we all have goals, and all of the CAN goals are just about the same. We want to help guide regional observation, research, and modeling to better understand localized drivers of acidification to our specific regions. We contribute to studies that will help predict consequences for marine resources and local communities, coordinate best practices for monitoring and data collection, and that also includes helping understand monitoring research and validation needs for MCDR. We help educate all of our stakeholders on coastal acidification's impacts and the solutions to acidification. And finally, we help to collaborate on mitigating and restoration planning within the Mid-Atlantic region. We are a partnership between MARCO's Mid-Atlantic Regional Council on the Ocean and MACAN. And if I mess this one up, Emma can help me out. The Mid-Atlantic Regional Association of Coastal Ocean Observing System. It's one of the IUCE associations. And it's also my pleasure to introduce Emma Venarde, who is our new MACAN co-coordinator from MACAN. So we're very excited that Emma has stepped into the leadership role for MACAN, and you will have a chance to see and hear more from her in the coming months. So I'm now going to throw it over to Emma, who is one of MACAN's program coordinators, and she will introduce our speakers today.
Thank you, Janet, and hi, everyone. So I'm really excited about this webinar, and I can tell from the turnout that you all are too. So we have first speaking as the director of the Nature Conservancy's Volgenau, Virginia Coast Reserve, Jill Bieri leads a staff of 14 working to protect, restore, and sustain the barrier islands, coastal lagoon, and bayside ecosystems of the eastern shore of Virginia. Jill holds a master's degree in marine science from the College of William and Mary's Virginia Institute of Marine Science, and she has over 30 years of experience working on the Chesapeake and coastal bays. So she'll be speaking first, very excited. And then next, Cassie Gurbisz is an associate professor of marine science at St. Mary's College of Maryland. Her areas of expertise are estuarine ecosystem ecology and undergraduate coastal science education. Her current research focuses on carbon and nutrient cycling in estuarine submerged aquatic vegetation systems. She earned her PhD in marine estuarine and environmental sciences from the University of Maryland in 2016 and has been active in the Chesapeake Bay science and education communities for nearly 2 decades. So thank you to both of our speakers for joining us, and I'll pass it off to Jill to start.
Great, thank you all. I'm excited to be here and I appreciate the invitation. I will say that I represent a huge collaboration, and I'll talk so much more about that as we get into the presentation. And I have the opportunity to bring together this collaboration into this market project, and really I'm excited to share it with you. A lot of times we do these presentations together as partners and collaborators, and so I hope that I represent all of our partners that are working on this in a way that makes everyone proud. So I'm going to talk about the Virginia Coast Reserve and why this is a place where we can think about implementing the first ever blue carbon seagrass market project, and really highlight the enabling conditions that exist at the Virginia Coast Reserve that answer those questions: why here and why now? And I always like to get started of, where in the world are we, just in case there are people who aren't familiar with where the Virginia Coast Reserve is. Of course, we are the southern portion of the Delmarva Peninsula, that Virginia portion of the Delmarva Peninsula, with the Chesapeake Bay to the west and the Atlantic Ocean to the east. And we're specifically talking about, so this is our mainland peninsula and these coastal bays behind the barrier island system off the coast of Virginia. So the seagrass restoration that we'll be discussing is really in this southern portion of these coastal bays of Virginia. And I'll talk about those barrier islands in a little bit. So it's really here in these coastal bays of Virginia where the largest successful seagrass restoration project in the world exists. A little bit of history about our eelgrass. It disappeared from this system in the 1930s, really along with eelgrass along the entire East Coast, mostly due to a pandemic, a wasting disease, and left our coastal bay system devoid of seagrasses. And then, of course, all those benefits that the seagrasses bring to the system. The isolation of this place, you can see there's really no direct connection to other places that might have seagrasses like eelgrass. And so there was no seed source after the wipeout of the eelgrass for it to come back naturally like it did in other places along the East Coast, including the Chesapeake Bay. So basically, the Virginia Coast Reserve was without eelgrass for over 70 years. And since the Virginia Institute of Marine Science began the restoration effort in the late 1990s, we have gone from basically zero eelgrass to over 10,000 acres of eelgrass, and it continues to expand. Just a reminder that seagrass meadows store about 2 times more carbon than terrestrial soils, so very important for carbon sequestration. And if we think about it over time, more than 5,000 metric tons of carbon have been stored in this seagrass that's been restored in the Virginia Coast Reserve. So why has this restoration been such a great success here? And why have we chosen now, and when I say now, in the last few years, to demonstrate the Blue Carbon Market Project here? So that's what I'm going to try to answer by talking about some of our enabling conditions. So there's a huge legacy of conservation along the Virginia Coast Reserve in this peninsula. 133,000 acres are protected. You'll see the ownership of these protected and conserved lands in the different colors. And, for more than 5 decades, our state and our federal and local partners, along with the Nature Conservancy, have been really working to protect this area. These barrier islands, the coastal marsh systems, and even places on the bayside here on this peninsula. So this is the largest stretch of coastal wilderness on the east coast of the US. And with that comes also a very low population of people that live in the Virginia Coast Reserve. And so we have very little runoff, very little sources of runoff that could provide eutrophication or anything like that. We have amazing water quality in these coastal bays. We have this exchange with the Atlantic Ocean, and so it's good water that's exchanged, and lots of good water quality. It's pretty amazing, actually. So a legacy of conservation is definitely an enabling condition for the success of these eelgrass restoration. Uh-oh. I can't get it to go forward. Let me start. Sorry about that. Okay, sorry about that. Always technical issues. So this is where we talk about the enabling condition of this extensive collaboration and partnership on a grand scale. So even, from the very beginning of this restoration project through this entire Blue Carbon Market project implementation, this collaboration and partnership has existed. VIMS, with funding from the State Coastal Zone Management Program, took the giant leap and started the restoration in the 1990s. UVA leads and coordinates much of the research, and I'll talk a little bit more about that. The Nature Conservancy's both global programs and our Virginia programs work together to steer the blue carbon process on this local to global effort. And of course, these seagrasses are on the bottom that's owned by the Commonwealth of Virginia, and they're the managers of these resources. And TerraCarbon cannot be forgot. They have been a huge technical partner as we've really entered into the feasibility through the verification and validation stages of this blue carbon project. It is here that we've also developed an innovative restoration technique. So we've really for this place determined the best way and the easiest and really the cheapest way to restore eelgrass is through seed dispersal. And so the process begins in the spring. In fact, we just ended this process a couple weeks ago, where we go out and collect intact reproductive shoots with the seeds still on the plants. We bring them back to our flow through seawater system. We put them in the tanks where they stay throughout the entire summer. The decaying vegetative material is pulled off and seeds fall to the bottom. And this sort of seed slurry that we have, these tanks have to be stirred every day. It's pretty labor intensive throughout the summer. But at the end of the summer into the fall, we have this slurry of seeds that can then be taken out and hand dispersed, hand thrown from boats. And that is our innovative restoration technique that has been working for several decades here at the Virginia Coast Reserve. We're pretty fortunate at VCR also to have probably one of the most studied coastlines in the world. And a lot of that is due to the University of Virginia's long-term ecological research here, funded through the National Science Foundation, where they've been looking since the mid-1980s at the effects of climate change on this intact barrier island system, including before the seagrasses were here, during and through this restoration. In addition, they've also done the first full carbon accounting for a seagrass meadow here, and they've developed the seagrass blue carbon methodology that we've used for this project right here. In addition, the Virginia Institute of Marine Science has been, as I said before, leading the restoration effort, coming up with this innovative planting technique, and they are responsible for the annual monitoring of all submerged aquatic vegetation, both in our coastal bays, but also in Chesapeake Bay, so that we really know how these grasses, the different species, are expanding or not on an annual basis. And that's really important for this project as well. It's pretty important to talk about in this project the political will and state level support. As in 2019, as we began the feasibility study for the market project, we really started thinking about the state ownership of the bottom, and was the Commonwealth of Virginia actually authorized to own and then sell carbon credits. We engaged with, at the time, William and Mary's Coastal Policy Center, and determined that we really needed legislation that would allow this project to proceed. And so during the 2020 General Assembly session, legislation passed and provided the authority for this blue carbon project to move forward. It basically says that the Department of Environmental Quality is the agency that can participate in the carbon markets for submerged aquatic vegetation in Virginia. It also specifically says that the revenue, any revenue resulting from the carbon sales, would go back into the monitoring and research and any administrative cost for this restoration. And it also allows the Department of Environmental Quality and the Commonwealth to enter into agreements for implementation. You can probably guess that with the state ownership and all of these partners working in collaboration, it's a pretty complex project to think about. And so we did develop a cooperative agreement with the Commonwealth of Virginia, we the Nature Conservancy, that acknowledges DEQ's role in protecting and enhancing the environment of Virginia. It acknowledges the Marine Resources Commission's jurisdiction over these bottomlands. And it talks about the Nature Conservancy's experience working to develop blue carbon projects, but also we have a lot of experience with terrestrial carbon projects. We all agreed to maintain and enhance the grasses that are there and the species that it supports and the water quality, which is all mutually beneficial to all of our goals and missions. So this cooperative agreement really allowed a chance for us to really define what each partner's role in the market project is. It also defined the project boundaries. So the blue carbon market project is just a small portion of the seagrass restoration project, and it allows for periodic review of that boundary. If it grows, if we move to different areas in the coastal bays, and this cooperative agreement is in effect, which is kind of unheard of for 30 plus years. I can't not list community engagement as an enabling condition for, and the importance of that for this project. We have engaged hundreds, if not thousands, of volunteers over the last couple decades to really help with that seed collection in the spring. And what happens, as everyone knows, if you engage members of the community and volunteers and really the greater community, it's not just people who live on the Eastern Shore of Virginia. They become great ambassadors for this project and the value of this project. And they've been a really critical element in the seagrass restoration. Just a couple things about the project. Our voluntary carbon market standard is the verified carbon standard. The methodology is listed here, which I will tell you is for tidal wetland and seagrass restoration, which does add to the complexity. Seagrasses are very different than tidal wetlands, and we found that out as we're going through this validation and verification project. The project proponent, in other words, who owns these credits, is Virginia DEQ. It establishes the start date as October of 2015. So we have in the project area where we know that seeds that were planted are actually contributing to this project, and they're expanding from those seeds that were planted, not from expansion from other areas. And the crediting period for this project is 30 years. Just a couple high level to end with the carbon project cycle. For those of you who might not be familiar, that feasibility stage that I talked about in 2019, as we begin to think about validation and verification, the design of the project design and description, the monitoring, how will this be monitored? How will we make sure that what we have established is actually happening out there? There's issuance of credits, and then there's the sale of those credits. specifically for this project, the development timeline, I've talked about a lot of it, but it's nice to sort of get it in a timeline. The project start date is when we can actually say in the project area where we start the project is October 2015. The feasibility study, that development of the project description and the monitoring plan for those years. We, in 2021, did that state review and approval, the agreement on the carbon rights that we talked about, and the coordination of that project boundary. And since 2022, we've been working on the project validation and the first verification, which would generate the first credits for sale. And in that process, it's very complex. It's the first one, Seagrass Blue Carbon Market Project. It's also, we're learning as we go, for sure. It takes a lot, a third-party validator that has to come out and do a site visit, provide findings. We have to respond to findings, and that really took almost 2 years for that to happen. Lots of reasons why that happened, but we can talk about. And then we're right now waiting for still waiting for the validation and the first verification that should happen by the end of this calendar year. And I know we're going to save questions until the very end, and I will stop there and thank you all.
Should I just jump right in?
Yes, you're good to go. Thank
All
you.
right. Share my screen. Yeah, so thanks everybody for coming today and for having me. I'm going to talk about a project. It's an NSF funded project that I've been working on that is looking at how SAV, or submerged aquatic vegetation systems, and estuaries affect the inorganic carbon system and how those interactions might affect the blue carbon potential of SAV beds. So I'll start by giving you a little bit of background about what's motivating this project, and then I'll tell you how we did the project and some preliminary results, but I don't have many results to share, so it's more just about kind of sharing what we're working on. So I'm sure everyone's familiar with blue carbon ecosystems. They're really productive coastal systems that remove carbon from the atmosphere and ultimately bury some of that as organic carbon in the sediment. And tidal marshes, seagrass meadows, or SAV, submerged aquatic vegetation, and mangroves are all really effective blue carbon systems. And my area of expertise is SAV, or seagrass. And so, yeah, in estuaries, we usually call it seagrass SAV because there's lots of different species that are included, but they're functionally pretty similar. So if we zoom in on the carbon cycle in SAV systems, we have CO2 gas diffusing into the water and forming dissolved CO2. And the plants are going to take up that CO2 through photosynthesis and fix it as organic carbon in their tissue. And then some of that carbon is then buried in the sediments. And especially in aquatic systems, the sediments typically anoxic and the decomposition rates are pretty slow. And so if that carbon stays buried in the sediment for long enough, it could be considered sequestered or removed from the atmosphere. And there's been a lot of research on these processes. So, Jill is kind of alluding to some of that work. The research has focused on how fast the carbon is buried and how much carbon is in the sediment, so carbon burial rates and carbon stocks. But in aquatic systems, there are a bunch of other forms of carbon, of inorganic carbon, that are in equilibrium with this dissolved CO2. So when CO2 dissolves, or it diffuses into the water, it's just because of a concentration gradient. So if the CO2 concentration in the water is less than the concentration in the air, it goes from the air to the water. And if it's higher in the water than in the air, it's the opposite. It goes from the water and out into the air. And when CO2 mixes with water, it pretty quickly forms carbonic acid. That's H2CO3 here. And then carbonic acid dissociates into bicarbonate and carbonate, and it releases these hydrogen ions along the way. Okay, so there's kind of this equilibrium, or all these different carbon species that just chemically can form depending on the conditions. And you can also move in the opposite direction along this kind of continuum here. So if there's a lot of free hydrogen ions in the water, the carbonate and the bicarbonate can accept those protons. And so systems that have a lot of bicarbonate and carbonate can accept a whole lot of hydrogen protons. And yeah, without driving the system all the way back into this CO2 phase. So, and also I'm stumbling a little bit over this because I am not a chemist, I'm an ecologist. So I'm working with a whole bunch of chemists that have really helped me learn about this system, but I apologize in advance if I'm oversimplifying it for some of you, if you're more chemistry minded. So the main point of showing this, I guess, is that the more carbonate and bicarbonate you have in the system, the more CO2 can ultimately diffuse into the system. So it's like sometimes called a solubility pump. So the more CO2 can come in and be stored in the ocean or in aquatic systems. And this is a closer look at the equilibrium between these different inorganic carbon species. And they're also related to hydrogen ion concentration, which is the same thing as pH. So pH is the negative log concentration of hydrogen ions. And so generally, when you have a higher pH, looking at the relative abundance of these different inorganic carbon species, you have, so at a higher pH, you have more bicarbonate and carbonate. And as the pH goes down, you have more hydrogen ions in the system. There tends to be more CO2 relative to carbonate and bicarbonate. And you can measure the net effect of the changes in this whole inorganic carbon system using a measure called total alkalinity, or TA, we abbreviate it by. So alkalinity is, there's lots of kind of complicated definitions of it, but what I'm going to go with for this talk is it's the balance of proton acceptors over proton donors. So proton donors would be these hydrogen ions in the solution, and then the proton acceptors are mainly the carbonate and the bicarbonate, which can accept those hydrogen ions. And so you can also think of it as the capacity of a solution to accept those protons, or like the size of the hydrogen ion sponge. And ultimately, the reason why we care about this in terms of blue carbon systems is because the higher the alkalinity, or the greater the alkalinity in the system, so the more bicarbonate and carbonate you have, the more the water can accept those hydrogen ions and therefore ultimately absorb more CO2. And so in SAV systems, there are a few processes that can affect the balance of that inorganic carbon system and therefore ultimately affect how much CO2 is going to be taken up by the water. So one of those processes is the SAV beds, they're often habitat for calcifiers like snails and clams and little tube worms. And so those calcifiers so there's that. And then there's sometimes in SAV systems, they can drive up the pH so high that calcium carbonate will precipitate out on the surface of the leaves. And so in both of those cases, we have calcification happening. So we have calcium ions combining with bicarbonate to produce calcium carbonate and CO2. So when calcification happens, CO2 is increasing in the system. And if the concentration of PCO2 in the water is greater than the atmosphere, it's going to diffuse out of the water. And then also at the same time, we're decreasing alkalinity. So because we're consuming the bicarbonate, the alkalinity is going down, which also kind of affects the CO2 sink capacity of the water. On the other hand, another process in SAV beds is that there tends to be a lot of anaerobic respiration because we have all that organic matter accumulating in the sediment, and that tends to aerobic respiration consumes the oxygen really quickly, and then the microbes start using anaerobic pathways to respire. And so there's a lot of different anaerobic respiration pathways that can happen, but a lot of them consume hydrogen ions. And so that increases alkalinity. And so if alkalinity is increasing, that could potentially increase the CO2 sink capacity of the water. And then one last process that might affect alkalinity in SAV beds is the assimilation of dissolved inorganic nitrogen. Just like, you fertilize plants, they need nitrogen in your garden. SAV also take up nitrogen. And they can use either nitrate or ammonium as a nitrogen source. So if they assimilate nitrate, nitrate is a negatively charged ion, so it's NO3 negative. And in order to maintain a neutral charge balance within the cells of the plant, they have to either extrude the hydroxide ion or take in hydrogen ions, which are positively charged. So if they assimilate nitrogen, basically that process is going to increase alkalinity, which could decrease the CO2 sink capacity. And then on the other hand, if they take up ammonium, they have to release hydrogen ions into the water or take up hydroxide ions. And so that's going to decrease alkalinity. So the effect on the CO2 sink capacity of the water in this case is going to depend on whether the plants are using nitrate or ammonium as their nitrogen source. So this is all, kind of complicated background information, but the point is that these processes aren't nearly as well studied as the organic carbon cycling in SAV beds. And there's some studies that are starting to show that these processes can be just as, if not more important than organic carbon burial in the ultimate source sink dynamics of SAV beds. So it's really important for blue carbon science to get a better understanding of, what's going on here. So where, when, how much are these processes affecting the carbon sequestration potential of seagrasses and SAV? I mean, part of the reason why we don't really measure it is because it's a huge pain in the butt, which I can tell you a little bit more about in a little bit. But it's also just, it's kind of just a new area of research. And this project is part of a much bigger NSF-funded study to understand how not just SAV, but other macrofauna or macrobiota in estuaries affect the inorganic carbon and alkalinity dynamics of estuaries. So we're looking at tidal wetlands, benthic fauna like bivalves and other clams and other organisms that live in the sediment, and SAV. And a bunch of chemists are doing really high quality characterizations of the water chemistry in the estuary. And this is a big multi-institution NSF project. So my part looking at the SAV effects on alkalinity are just kind of a smaller part of this big project. And we are working in the Chesapeake Bay. So here's the Chesapeake Bay. We've got New Jersey, Delaware, Maryland, the Appalachian Mountains. And, it's very convenient for me because I live right here in St. Mary's County, so right on the bay. But also the Chesapeake Bay has a bunch of tidal tributaries, and the alkalinity in those tributaries tends to encompass the range of alkalinities that we would expect to see in estuaries around the world. So we've got the Potomac River, which has the highest average alkalinity out of all the tidal tributaries. And then we also have, the Pamunkey and the Mattaponi down here, which converge into the York, have the lowest alkalinity in the range of alkalinities. And so these are really nice kind of end members to use to study how alkalinity is affected by the macrobiota in these estuaries. And so, so for my part of the project, the question I'm asking is how do SAV beds along estuarine salinity and alkalinity gradients affect alkalinity fluxes? And I'll just tell you right now, I don't know the answer. We don't really know yet. The rates are really variable, which, because it's so biogeochemically complex, it's not surprising. But we're also still processing a bunch of our data, so we don't even have a full data set to look at yet. But what I will show you is how we measured the fluxes in SAV beds of alkalinity. I'll share a few preliminary results and then talk about what we're currently working on. So for my SAV study sites, we had 4 sites. So we were working in the freshwater and brackish areas of both the Potomac and the York River. And these are, you might see these abbreviations in some of the other slides that I'm going to show. So FW is freshwater, BR is brackish, POT is Potomac, YOR is York. And we measured the rates of change or the fluxes of alkalinity and a bunch of other parameters in SAV beds using in situ flux chambers. So we went out and deployed these chambers in the grass beds with 6 different treatments. So we had chambers encompassing SAV and sediment. We had chambers just with bare sediment, and then chambers with water only. And that's so that we can tease apart the effects of, SAV, the sediment, and the water, and, how they're interacting with the inorganic carbon system. And we also had clear chambers and chambers that were covered basically in dark trash bags to replicate dark or nighttime conditions. And we incubated the chambers for up to like 5 or 6 hours, depending on the time of year. And we did this 3 times a year. So spring, summer, and fall. So that's 4 sites 3 times a year. That's a lot of field work. And this is what the chambers look like when they're deployed in the field. So they're just made out of PVC, and we have this clear vacuum packing bag here that like dissolved gases can't pass through. And then a sampling chamber, or sampling port here, where we deploy the chambers. We would use a syringe to withdraw a water sample at the beginning of the incubation, and then withdraw another sample at the end of the incubation, and then use the difference in those 2 measurements to calculate the rate of change in whatever parameters we're interested in. So alkalinity is what I'll talk about here. And then I also put pH and oxygen loggers inside the chambers. We also collected the SAV biomass from all the chambers that had SAV in them, and we kept any animals or shells or organic detritus that was greater than 1 millimeter in the SAV and all the sediment. And then we also scraped the SAV leaves for calcifying epiphytes. And actually, like at one of our sites, they had quite a bit of, these are little actual tube worms that have calcium carbonate shells that are growing all over the SAV. And this is what a SAV biomass core looks like. Okay, so I'm just gonna kind of briefly walk you through some of the results and kind of speculate about what they may or may not mean, and then we'll just and then we'll wrap it up. So the first results I want to show are, these are the dark rates of alkalinity change. And I wanted to look first at just like how the rates in the benthos are different from the rates in the water column. So I have my SAV and the bare sediment treatments combined here, and we're comparing them to the water column alkalinity flux rates. So we've got alkalinity flux, we've got our 4 sites, and we've got the different seasons on the x-axis. And this line going through 0 is indicating, so at At 0, there's no change in alkalinity. If the numbers are positive, that means alkalinity increased. So that means there's alkalinity production happening in the system. And if it goes down, then alkalinity is decreasing. So there's alkalinity consumption in the system. And so here's a little cheat sheet sort of summary of the processes that I talked about in the introduction. And then these arrows are the difference. I'm basically eyeballing it at this point. I haven't done any kind of rigorous stats on this data yet, but I'm just looking at whether these benthic treatments, there was an increase or decrease in alkalinity relative to the water column treatment. So, is the sediment and the SAV having an effect on alkalinity? And so it's kind of variable, but in most of the sites in the seasons, there seems to be an increase in alkalinity in the sediments. And to me, so that makes sense because in the dark, you have aquatic sediments, there's probably anaerobic respiration going on, and that's going to increase the alkalinity. Okay, so now if I zero in on the SAV treatments in the dark versus just the bare sediment treatments in the dark, we can start to look at, what kind of effect is SAV actually having on these processes. And again, I'm just eyeballing here, but it's a lot more all over the place. Sometimes there seems to be an increase in alkalinity, sometimes there's a decrease, sometimes there's not really any difference, or the rates are just so variable that it's hard to tell if there's a difference. But in the cases, okay, so yeah, these are lots of I don't knows. I don't know what's going on here. There are a few cases where it does seem like alkalinity increased in the SAV treatments. So what could be going on here is just there's more biomass. So even like the live biomass is respiring in the dark. And, probably in these treatments, there's more organic matter in the sediments. So we could have more organic respiration going on, increasing the alkalinity. And then, sometimes SAV, they can leak oxygen through their roots, even in the dark, like, if it's not long after the sun has been shut off. And so we could have the SAV just kind of increasing the aerobic respiration, turning off some of the anaerobic respiration in these treatments, or there could be calcification going on. There could just be more calcifiers in these treatments. But again, like they're pretty variable, so it's hard to say. Okay, so now we're going to show you some of the alkalinity fluxes in our light treatments. So here we're looking at, and I'm not even looking at the water in these, we're just kind of going to hone right in on the SAV versus the bare unvegetated sediment treatments. And so it's, again, it's pretty variable. There's not as many question marks. Like it does seem to be like the SAV is having effects, but there doesn't really seem to be any kind of consistent pattern in those effects. So in some cases, again, we have alkalinity maybe increasing, or in some time definitely increasing a lot. And that could be nitrate assimilation. Although I was in this particular, at this site, there's also a lot of this filamentous cyanobacteria called Microcera wolii that could also be doing something weird. I don't know. So we have, in some cases, a small or big increase in alkalinity because of the SAV seem to be having that effect. And then in other cases, it seems to be decreasing. So it could be a calcification, or possibly the plants in these experiments are using ammonium as their nitrogen source. And just like one other thing I wanted to point out is that, so in general, the biggest SAV effects seem to be in the light treatments, which makes sense because in the light, the plants are photosynthesizing. They're just more metabolically active. And then, sort of spatially, the biggest effects are in these freshwater sites. But it's interesting, in the Potomac freshwater, there's a clear increase in alkalinity, but in the York site, there's, at least in the summer, there's a big decrease in alkalinity. So yeah, I don't know if it has to do with the fact that this one has that microcera cyanobacteria and this one doesn't, or it has to do with the underlying water chemistry. That's something we still have to dig into. So yeah, like I said, I'm not going to tell you a whole lot. That's like pretty much all the results that we have. I actually just got a bunch of the data from our field sampling from 2024 in, so we're still processing the data, but what we do know is the flux rates definitely vary seasonally and across, and even within sites. SAV sometimes has an effect, sometimes it doesn't. The largest effects seem to be in those freshwater zones. So we're continuing to work on the data. We also have, for a subset of our samples, calcium ion and nutrient data. So we can look at the fluxes of calcium to determine whether calcification is happening or calcium carbonate dissolution. And we also have nutrient data to look at fluxes of nitrate and ammonium to start to kind of tease apart those mechanisms. And ultimately, we want to work towards being able to estimate the effect of these alkalinity fluxes on PCO2 in the water, and kind of make some inferences about whether and how much these effects might these interactions might affect the ability of the water itself to absorb carbon dioxide, and whether that might offset any of the blue carbon that's buried via organic carbon burial in these systems. So yeah, thanks for listening. And I just want to acknowledge all the field help and field help I had, and the help that I got from analyzing samples and our collaborators. So yeah, thank you.
Well, thank you to both of our speakers, and Cassie, it sure looks like you had a lot of fun in the field. So we want to open it up now to questions. If anybody has any questions, you can either use the chat or the Q&A. And so I have one question for you, Jill. If you mentioned that you like to get the community involved in your seeding or seed collection. Do you have a specific program that community members volunteer for, or how do you engage folks around the seeding?
Yeah, sure. Thanks for that question. So we have a larger community engagement program at the Virginia Coast Reserve, and one of the activities that we open up for volunteers, anyone who wants to, contribute to the effort, is the seed collection. It requires people to be comfortable in a boat, in a wetsuit, in chilly water. It's usually not putting their head underwater, but we typically have a waiting list for those volunteer opportunities. Yeah.
Wow. Even though you already have your waiting list, would you mind sharing with us the any information? We'll be happy to send it out to the community and get you an even larger waiting list.
Sure. Yeah, I'll share our volunteer coordinator who sits in our Charlottesville office, and she provides opportunities to really great ways for volunteers to get involved. There's the typical things that you would think about for volunteering, but building oyster castles, or collecting eelgrass seeds, or helping steward our barrier islands for the migratory birds. Like, we have some really cool opportunities. I'd be happy to share that information with you.
That's great. Thank you so much. Anyone else have any questions? Looks like we do have one in the chat. Does Zostra typically prefer the NH4 or NH3, or is that even known?
It might be known, but I don't know. And it also might depend on, it might just depend on which one's more abundant. But I haven't dug into that in the literature, although I'm sure that literature does exist.
Thank you. Give it another second to see if anybody else pops up for questions. Here we go. For Jill, you mentioned your process for collecting / maturing the SAV seeds. Does this involve turbulating?
Well, I guess I need to understand what turbulating is because I don't know what that means. The process is we collect the seeds on the reproductive shoots. Those reproductive shoots are put in those flow through tanks that have flow through seawater. We stir them by hand, by feet mostly, people walking around in the tanks to keep, to keep the oxygen in the tanks and to keep the decaying materials sieved out and stirred up. And those seeds fall to the bottom, and those seeds, by the end of the summer are sort of a slurry. And that's where we can start to take the tanks and combine them and have all of the seeds in one tank, and then get an idea of how many seeds we have. And then they're held and put out in the system in like early October.
Well,
What does turbulating mean? Does anyone else? Okay, thanks. I don't know.
And I could have been mispronouncing it. I'm not sure.
Yeah.
And when people restore some estuary and SAV, some people at Anne Arundel Community College developed a system where they blast the tank with air and turbulates the water and kind of forces the
see.
reproductive material to get separated from the seeds.
Yeah. This happens over time. It's a longer process. Yeah. Thank you.
Another Delaware is apparently in the process of getting a turbulator going as well. So that's great. It's wonderful to have more of these programs developed. Another question we have: has all of your work been performed with cultivated seeds, which appears to be successful, or have you considered repatriating rhizomes? I guess transplanting rhizomes.
So, this is the technique that works in the coastal bays and in Chesapeake Bay for eelgrass. And all those like the enabling condition of really good water quality and the correct sediment and all that. And for years, for decades even, different techniques have been tried with eelgrass. And one of those was transplanting whole plants. There's been all kinds, and I did that as a graduate student, in the early 1990s. So we really have tried a lot of those different techniques. And this is the technique that works for eelgrass seeds in the coastal bays and in the Chesapeake when there's good water quality. So, it's important to say that just because that's what works here doesn't mean that could be replicated in other systems with other species. But, we have found in this system that has really good water quality, this is a really simple way to do it. It takes a lot of space over the summer and a lot of manpower over the summer, but it is something that, we basically can do pretty economically.
Also, another comment in the Q&A: Delaware has a turbulator that has been in use the last 2 years and is in the process of building an updated one. Mainly involves pumping air into the tank. We use shop vacs through a PVC system and toss the plant material on top of the jetted water to release mature seeds that will fall into the bottom of the tank for collection. It's great. Thank you all for sharing your experiences with seeding, because it sounds like this is a very successful method, and we look forward to hearing more results in the future. And for those of you that contributed to that conversation, oh wait, it's got another one. Should collected seeds be buried or cured before seeding?
So we call the holding in the tanks the curing of the seeds. They're not typically buried. They fall to the bottom. They're neutrally buoyant. So when we throw them out of the boat, we use planting patterns and grids so we know where to come back and monitor for success. But the seeds are not placed in the bottom. We are working with a company right now to look at that, like how, what would it be like if we injected the seeds in the bottom? Would there be more success? But right now, the way that we do it, we cure them over the summer and hold them in these tanks, and then they are ready when the water temperature falls in the fall, because they're a cold water species that germinate in colder water conditions. And so in the fall is the best time to disperse the seeds, and then they begin to germinate.
And another comment we have is the success in planting cultivated seeds is remarkable. We're starting to try to expand eelgrass meadows in New Jersey Barnegat Bay. So looks like we're spreading the word.
It's awesome. That's great. Good to hear.
Yeah. Well, and then another question, how many seeds are collected, or how are the seeds collected? Sorry, how are the seeds collected?
So the volunteers and some others will go out and collect the reproductive shoots when the seeds are just about ready to be released from these reproductive shoots. We want these seeds to be released in the tanks throughout the summer. And so we basically hand harvest them with snorkels and then bring them back and put them in the tank. So they are hand harvested, the reproductive shoots. And they're pretty easy to tell if you've ever been in an eelgrass bed. The difference between, there's a little bit of a learning curve when volunteers come out to understand what they're collecting, but they look very different than the vegetative shoots of the eelgrass, especially when the seeds are intact.
Very interesting. All right, well, it looks like we are out of questions and just about out of time as well. So we want to thank everybody for joining us today, and hope that you'll pass the recording of this webinar along, and we look forward to connecting with all of you again in the future. Thank you.
Thank you, speakers.
Thank you. Appreciate it.
Yeah, thanks for having us.
Yeah.